Welcome student. Today in another lecture we discuss about the scope and importance of biotechnology. In the previous lecture we discussed about what is biotechnology. To a glimpse, we remember that biotechnology is the integration of biological sciences and engineering sciences in order to achieve the application of organisms, cell parts, part thereof, and molecular analogy for the product and services. According to the International Union of Pure and Applied Chemistry, it is the application of biochemistry, biology, microbiology, and chemical engineering to industrial process and product and quality. Just brief about what is the process of biotechnology, how we can achieve the biotechnological product. There are certain six steps in biotechnology. First step is the isolation of DNA from the donor organism, followed by DNA fragmentation using certain specific restriction endonucleases. These endonucleases have the property they can cut the DNA at specific recognition sequences, which is either 6 base pair in length, that is called the restriction endonuclease. Afterward, the ligation of the desired fragment into the vector can be done. This vector is a delivery vehicle that allows the transportation of that material into the specific host. The recombinant DNA is then developed through this process of binding of this vector with the restricted DNA fragment is then transferred to the host cells. In normally we can use E. coli as a host. Then culture of transformed cell in a nutrient media for their large scale multiplications and followed by extraction of the desired product. Now, if we talk about the principle of biotechnology, the principle behind the biotechnology is based on two terminology, that is first is genetic engineering or recombinant DNA technology and followed by bioprocess engineering, that is called downstream processing. Now, talking about one by one, first we talk about the genetic engineering. The genetic engineering is the manipulation and modification of the genetic material in an organism or plant to insert desirable traits. Recombinant DNA technology is an technique to alter the gene of an organism. This desired gene is inserted into the host cells, and after that we can identify the phenotypic expression of that particular gene in the host system. This recombinant DNA technology involves the following steps. This first step is the selection of the desired gene. Second, selection of vector for the transfer of gene known as cloning vector or plasmid host. The common is plasmid. We have plasmid, we have bacteria, etc. Then we have the insertion of recombinant DNA into the host cells, followed by maintaining the introduced DNA in the host cells so that it is passed on the next generation. Now, this is the diagrammatical representation of process how this recombinant DNA can be produced. First, we have the plasmid working as an extra chromosomal DNA in the bacteria. We isolated that plasmid, then cutted this plasmid with the help of restriction endonucleases, and then the specific gene of interest which we want to introduce into the host system, we can cut that DNA with the same restriction endonucleases and then ligate it with the help of DNA ligase. Here we have used the human insulin gene for the recombinant DNA formations. After the recombination DNA ligations, we can introduce this into the bacteria. The process is known as transformation. And after transformation, we can go with the multiplication of bacterial cells to increase the production of insulin genes. Here you can see the dotted lines, that is the production of high quantity of insulin with the large number of bacterial cells having the recombinant DNA. Similar to the recombinant genetic engineering process, the another process is the bioprocess engineering. The modern biotechnology is responsible for the advancement in the pharmaceutical industry. It has the production and storage of products like antibiotics, enzymes, vaccine at large scales. A large amount of culture can be obtained by carrying out multiplication of organisms in the bioreactors under sterile and optimum conditions. We get a high yield of the required product using this bioprocess engineering tools. This bioprocess engineering will follow the different processes like we can obtain the product that is subjected to a series of processes before being marketed. Then products are purified by a process called downstream processing and formulated by undergoing clinical trials. Then the product undergoes strict quality check before it is subjected to the further trials. This is a flow diagram of bioprocess engineering. Here we can first of all we prepare the biomass, that is called inoculum stage. Here we can edit the raw material, which is a nutrient in the reactant that allow the growth of the microorganism or specific cell lines under aseptic condition, that is the sterile conditions. Here we have the bioreactor. Bioreactors are the chambers of area where we can increase the production in the twofold or fivefold in productions. After this bioreactor formations, we can go with the downstream process, that is the product recovery. Here we have different steps like cell separation, cell disruptions, product extractions, product concentration process, then product separation, purification, drying, and finally we got the specified product. Now talking about the branches of biotechnology, there are different branches inside the biotechnology. Based on your animal biotechnology, we have medical biotechnology, we have plant biotechnology, environmental biotechnology, industrial biotechnology, genomics and proteomics, bioinformatics, nanotechnology, etc. We can look a glimpse about these different types of biotechnology one by one. First, we talk about the animal biotechnology. As the name indicates, animal biotechnology, it deals with the development of transgenic animals. The transgenic animals are those animals which carry specific genes from other organisms that have the certain specific features that make them the important animals for increased milk or meat production with resistance to various diseases. It also deals with in vitro fertilization, that is most commonly known as the IVF technology, where we can allow the fusions of egg with the help of sperm in the artificial environment, and then this developed embryo is transferred to the surrogate mothers. This is the process how we can get it the IVF techniques and developing a clone here that we can remove the nucleus, insert it into culture cells. After electrophoresis with the cell culture, we are able to get it the cloned embryo, and this the clone profile is developed through this technology. The transgenics, as I already told you, that this is the recombinant DNA technology product where we can insert it DNA from another sources to a specific organism. Here this is the process how we can get it the recombinant organism. Like in this case, we can find that we have spotted mice development with the help of this recombinant DNA technology. The another area is the gene splicing. That gene splicing where we can splice out the gene from a host organism or targeted organism and lead to the development of another organism, that is called the donor organism, so that we can have this specific gene of interest. This is the process where we can develop isolated insulin gene from human cells, add it into the plasmid, making a recombinant plasmid. After that, insert it into transgenic bacteria, grow into the large population number with the help of culture media, and then we are able to get it the insulin product. The another area in the animal biotechnology is animal cloning. Animal cloning, as the name indicates, to make the identical ones. These are the genetically identical to the parent one. How we can develop the genetically identical offspring and what is the need behind to develop this genetically identical offspring? As the scientists are going with this multiple copies of mammals that are nearly identical copies of other animals, including transgenic animals, genetically superior animals, and animals that produce high quantities of milk or certain metabolites that are of various uses. Today, cattle, sheep, pig, goat, horse, mules, cat, rat and mice have been cloned, and starting from the first clone was developed in 1996, commonly known as Dory, and the process is known as somatic cell nuclear transfer method, SCNP. Here we can use the nucleus from the egg cells, two sides, and replace it with the nucleus from a donor adult somatic cells, and after the fusions with the sperm cells, we are able to get the embryo, and this embryo is directly transferred to the wound of the surrogate mother, and afterward the cloned animal is developed. This is the process how we can develop the Dolly sheep here. We can use this donor nucleus, and egg cell is coming from the mother, from where we enucleated the egg cell so that we can remove the nucleus, and then fusion, after fusion with the nucleus of the donor, we can make the fused cells, and embryo is developed, which is transferred to the surrogate mother to make the clone baby. Similar the process is coming in the diagram. We are able to fuse the enucleated cells, and then clone is developed. These are the some companies that relate with the animal cloning purposes or development of large scale production, like Alembic Pharmaceuticals, Alvira Animal Health Limited, Biotechnology, Natural Life Science, Ayurved, Delchem Biotech. These are going with the large scale production of certain specific metabolites like pharmaceutical products like animal feed, meat, urine, distilling, and so on. Now coming to the another technology, that is medical biotechnology. Medical biotechnology is your use of living cells and cell material to research and produce pharmaceuticals like enzymes, antibiotic, vaccine, etc. It deals with the diagnosis of various diseases, large scale production of various drugs and hormones such as human insulin, interferon, gonadotropin hormone, vaccines for certain pathogens like viruses, chickenpox, rabies, polio, growth hormones such as polio. And recently we praise about the pandemic with the help of, due to this COVID-19, and we are able to develop the vaccine against COVID-19 viruses also. These are the area where we have the plenty of works in the or scope in the fields. The most important in the area of cancer related conditions, followed by infectious disease, then autoimmune disorders, diabetes, cardiovascular diseases, where we have this approaches of this biotechnology, invading this biotechnology in the large scale production of certain metabolite and eradications of these type of disease from the society. These are the certain of the examples, like we have the hormone, follicle stimulating hormone, growth hormone, insulin, growth factors like platelet derived growth factors, nerve growth factors, insulin growth factors, cytokinins, vaccine, which are developed through the biotechnological process. Some of the biotechnological products, as the name human insulin, we have humulin, then somatotropin, that is humultrops, then erythropoietin alpha, uroafusine, then factor 8, bioclate, then we have interferon alpha 2, roperon A, and these can work against certain type of disease, and these are the companies which makes these type of products like Eli Lilly, Janssen, Roche, Scans, etc. Now talking about the third area, that is the industrial biotechnology. Industrial biotechnology is a set of practices that use living cells such as bacteria, yeast, algae, or component of cells like enzymes to generate industrial product and processes. It can be applied to food technology, it can have the role in beverage industry, it develop new process for higher productions of certain metabolites. It deals with the commercial production of various useful organic substances like citric acid, acetic acid, acetone, vinegar, glycerin, etc. Antibiotic like penicillin, streptomycin, mitomycin, through the use of certain microorganisms, maybe bacteria, maybe fungi. Then these are the certain some of the examples developed through this industrial biotechnological aspect. First is the biofuel production, renewable fuels. We have bioethanol, biobutanol derived from corn starch. We have biomass-based biodiesels, renewable diesels from soil or waste oil fats, grasses. We have advanced biofuels, ethanol from sugarcane. We have cellulosic biofuels, cellulosic ethanol, cellulosic diesel, etc. that are developed as a second generation ethanol. Then these are the microbial enzymes production to the industrial scales, like we have developed the alpha amylase, we have lipase, we have fungal lactase, pectinase. These enzymes are used in day-to-day life in the industrial process for development of certain specific type of product or making certain type of specific process. These are some of the companies working in India in the field of industrial biotechnology, like we have Biotechnology Limited in Bangalore, where we have large scale production of industrial enzymes. We have advanced enzyme technology in Thane, enzyme for human and animal healthcare, textile, leather and food grains. We have Concord Biotech in Ahmedabad, producing exporter of enzyme strains, having state-of-the-art fermentation facilities. International Panacea, New Delhi, work on product by fermentation process like biofertilizers, biopesticide, nutraceuticals, recombinant protein and biomolecules. We have Tex Biosciences in Chennai, where we manufacture enzymes using textile, leather, poultry, feed, stone washing paper and for dye. We have Praz Industries in Pune, a global ethanol technology provider who has also entered into biodiesel technology. Now, the next area that is the environmental biotechnology deal with the detoxification of waste, because we are facing nowadays the problem of pollutions. So this is the branch that allow the removal of pollutant from the environment. Industrial effluents, treatment of sewage water and control of plant disease and insect through the use of biological agents such as viruses, fungi, bacteria, etc. Some of the major application of environmental biotechnology includes biomarkers, biosensors, biofuels, bioremediations and biotransformations. If we talk about bioremediation, it's a process utilizing certain microorganisms for cleaning the environment, polluted either in the form of water, sediments or soils. To achieve this process, we are using certain type of microorganisms that will allow the biorestoration, biotreatment and bioreclamation of the water. Then we have the microbial enhanced oil recovery. Scientists apply different molecular approaches to encourage hydrocarbon utilization in oil wells. This purifies the oil, therefore reducing processing costs for increased profitability. Then bioelectrochemical systems. Here we can use a microbial electrolysis process. Microbial fuel cells are used to innovate the fuel production, reducing energy consumption during waste treatment. Bioelectrochemical system will generate adequate electrical energy for industrial or domestic uses. Similarly, industrial waste treatment can be done through the microbial biodegradation and detoxification of quality environments. That it allows the acceleration of degradation process to treat the large volumes of waste. Companies are now using bioaugmentation process to clean up the environment. These are the certain companies working on this area, like we have Anover in Bhubaneswar, microbial resource management. We have MS Green Solutions in Coimbatore. We have Terry in New Delhi, the Green Circle in Vadodara, Biofuel distributor in India and Convert in Mumbai, that clean up the environment by certain types of pollutant. They are developing certain types of technology, producing certain microorganisms, improve microorganisms, develop recombinant organisms to clean up the environment. Then we have another area that is plant biotechnology. It is a combination of tissue culture and genetic engineering process. Tissue culture is a process where we, under aseptic condition, we are able to develop a complete plant from a small piece of plant. In general, we can say this is the explant. Any living part of the plant can work as explant because of a phenomenon that is called totipotency. Because of this totipotency phenomena, plant have the capability that from a single cell or a piece of tissue, we are able to develop a complete plant. Here we have large scale production of haploid plant, abiotic and biotic stress resistant plant, embryo rescue methods, clone multiplication, clonal propagations, and cryopreservation process coming under this plant culture step. Then the main features of this biotechnology is here we are developing the herbicide tolerant plant. In these pictures we are developing the genetically modified form. So by these pictures, using genetic engineering tool of plant tissue culture technology, we are developing some improved variety to feed the nations, to feed the hunger people, so that we can develop the product in a large scale levels. Then these are the certain examples: genetically modified foods which eliminating undesirable food component. Plant biotechnology can reduce a number of undesirable food components, such as one of the major allergic protein in rice or the major allergens in peanut or soya bean. We can also afford to reduce or eliminate other undesirable component of food like glucosinolates in canola, wheat, protease inhibitor in beans, glycoalkaloids in potato, and mycotoxin in corn that can be developed through this genetic engineering approaches. Then coming to the nanotechnology. Nanotechnology is another area which is working at the nanoscale level. Nanoscale, what does nanoscale means? That a molecule that is at a size of 1 to 100 nanometer in range, that is called the nanoparticles. Why we are working on this area, nanotechnology? Because if we talk about the natural system, if we see that natural system, the butterfly, the beautiful wings of butterfly, they have the nanoparticle arrangement to make their effectiveness or attractive toward the pupils. So this type of features are already available in the environment. So these nanoparticles at the small scale level, they have the high surface area that allow the accumulation of certain compounds, that allow the targeted effectiveness, delivery of certain metabolite, delivery of specific drug toward the targeted, and these metabolite or nanoparticle can work against specific abiotic and biotic factors. Then we have mucosal vaccine developed through this plant biotechnology. Here we have the tomato. There we are developing genetically modified flavor saver tomato through this plant biotechnological approaches. These are the related companies working in the area of plant biotechnology, like Crop Domain in Bangalore, that work on the climate smart agriculture biofoods. Innovation contributes to the method of building plant size specific functional microbiomes or sampling defined beneficial traits of plant. Then Enverto Agro Foods Private Limited in Calcutta, working on biosensor, bioelectronics development. Then we have Caroline in Chennai, we have Chetnigri in Vadodara, working on disease diagnostic and development of a specific type of plant producing certain human nutraceuticals. Then talking about the scope of biotechnology, we have this biotechnology in different areas like working in the clinical research, medical science, environmental science, animal science, pharmaceuticals, microbiology, quality control, biochemistry, forensic science, toxicology, and pathology, where we can work on this area. Like when we have the certain type of organism that causes infection in human bodies, animal body, or in plant, so we can remove these microorganisms, we can kill these microorganisms. Or second approach is we can develop a plant or develop an animal that cannot allow the growth of these type of microorganisms in their body. So such type of features, we have the plenty of applications in the biotechnology. For this, by this we are ending our lectures today. In later on class we discuss about the future aspects of these biotechnology. Thank you very much.